नमस्कार विद्यार्थी मित्र, आज आपण समजून घेणार आहोत सेल्स ऑफ इम्यून सिस्टीम. आपली जी इम्यून सिस्टीम आहे, इट इज मेड अप ऑफ टू टाइप्स ऑफ cells. First type is lymphocytes and the second type is antigen presenting cells. So these are the two types of cells in our immune system. Lymphocytes are the main cells of our immune system. They are produced from the stem cells which are present in bone marrow of adults. Bone marrow of adults and liver of fetus. These stem cells differentiate or proliferate to produce the lymphocytes. Stem cells proliferates into lymphocytes, and that process is called hematopoiesis. So you should remember this term also. What is hematopoiesis? It is the process of formation of lymphocytes from the stem cells, and this process of hematopoiesis takes place in the bone marrow of adults and in the liver of fetus. लहान मुलांमध्ये ही प्रोसेस लिव्हर मध्ये होते आणि मोठ्या व्यक्तींमध्ये ही जी प्रोसेस आहे ती बोन मॅरो मध्ये होते. These lymphocytes are produced in the bone marrow, but their differentiation takes place, their maturation takes place at two different places. Some of the lymphocytes get matured in the bone marrow itself or in the basal lymphoid tissues. Those are called B lymphocytes. And the lymphocytes which are produced in the bone marrow but differentiated into the thymus, those are called T lymphocytes. So simply lymphocytes ज्या आहेत त्या आपल्या इम्यून सिस्टमच्या मेन सेल्स आहेत. त्या बोन मॅरो मध्ये तयार होतात, प्रोड्यूस होतात. त्या प्रोसेसला आपण हिमॅटोपॉइसिस म्हणतो. परंतु त्याचं डिफरन्शिएशन वेगवेगळ्या ऑर्गन्स मध्ये होतं. काही बी लिम्फोसाइट्स ज्या आहेत त्या बोन मॅरो मध्ये किंवा बर्सल टिश्यू मध्ये मॅच्युअर होतात, डिफरन्शिएट होतात. त्यांना आपण बी लिम्फोसाइट्स असं म्हणतो. आणि ज्यांचं मॅच्युरेशन किंवा डिफरन्शिएशन थायमस मध्ये होतं त्यांना आपण टी लिम्फोसाइट्स असं म्हणतो. थोडक्यात lymphocytes are again of two types, B lymphocytes and T lymphocytes. Now we will discuss the difference between B lymphocytes and T lymphocytes. B lymphocytes and T lymphocytes. B lymphocytes differentiation takes place in bone marrow or basal lymphoid tissue. That's why they are called B lymphocytes. These B lymphocytes, on getting sensitized, once they become stimulated or sensitized on stimulation, and they produce plasma cells. B lymphocytes stimulate झाल्यानंतर, स्टिम्युलेट केव्हा होतात? त्या अँटीजेन गेल्यानंतर, अँटीजेन त्यांच्या संपर्कात आल्यानंतर, राईट. स्टिम्युलेट झाल्यानंतर काय तयार करतात? प्लाझ्मा सेल्स तयार करतात. आणि त्या दिस प्लाझ्मा सेल्स प्रोड्यूस अँटीबॉडीज. प्लाझ्मा सेल्स काय तयार करतात? अँटीबॉडीज तयार करतात. These antibodies fight against the pathogens or antigens. Means B lymphocytes do not directly kill or directly fight with the pathogen, and that's why we can say B lymphocytes gives us humoral immunity, or we can call it antibody mediated immunity. Antibody mediated immunity. These B lymphocytes, they are very important in destroying of extracellular pathogens. The pathogens which are not in cells, they are outside the cells, they are present in the blood or lymph. To kill those pathogens, these antibodies produced by B lymphocytes plays important role, and that's why we can say B lymphocytes provides us antibody mediated immunity or humoral immunity. Now T lymphocytes. T lymphocytes, these are also our immune cells. They are of 4 types. There are 4 types of T lymphocytes. The first type is helper T cell. They are called TH. Second type is killer T cell. In short, we can call them TC. Cytotoxic killer cell is another name for this. Third is suppressor T cells, in short we write it TS, and fourth type is memory T cells, TM. So these are the 4 types of T lymphocytes which plays important role in our immunity. We will discuss all these 4 types of T lymphocytes in detail. Next topic is types of T lymphocytes. T lymphocytes are of 4 types: helper T cells, killer T cells, suppressor T cells, and memory T cells. First one is helper T cells. Helper T cell. They are also called TH. These are the cells of immune system which get sensitized first and make whole immune system alert about the invading pathogen. यांना जनरल्स ऑफ इम्युन सेल्स असंही म्हणतात. जनरल्स ऑफ इम्युन आर्मी, बिकॉज ह्या स्वतः अँटीजेनशी लढायला जात नाही, पण यांचं जे काम आहे, यांचं जे फंक्शन आहे, जे आता मी तुम्हाला सांगणार आहे, ते इतकं महत्त्वाचं आहे की ह्या एखाद्या सैन्याचा जसा कमांडंट असतो किंवा जनरल असतो, एखादा अधिकारी असतो, तो जसा निर्णय घेतो, तसं ह्या ज्या हेल्पर टी सेल्स आहेत त्या स्वतः युद्धामध्ये भाग घेत नाही, स्वतः लढाईमध्ये भाग घेत नाही, पण त्या इतर सेल्सला स्टिम्युलेट करतात. सो सेन्सिटाइज्ड हेल्पर टी सेल्स कॅरी some functions. They stimulates B lymphocytes for the production of antibodies. They also stimulates the killer T cells for the enhancement of phagocytosis. These killer T cells are phagocytic in nature. They starts proliferation of immune cells. And how they do this? they gives chemical signal. This sensitized helper T cell gives chemical signals to B lymphocytes, to killer cells, and due to that, those get activated or stimulated. And those chemical signals produce chemicals, lymphokines. Those chemicals are called lymphokines. They produce lymphokines, and through these lymphokines, they gives chemical signals to B lymphocytes and other immune cells, and they stimulate them. Second type is killer T cells. They are also called cytotoxic T cells, or in short, they are written as TS. As the name indicates, they are the killer cells. They destroy or kill the pathogen, or you can say antigen, or you can say cancer cells. They also kills cancer cells. How they kills these pathogens or antigens or cancer cells? Suppose this is the antigen infected cell. These killer T cells secretes chemicals called porphyrin, and these porphyrins, they make holes in the membrane of infected cell, and due to that, this infected cell get killed or destroyed. So in this way, these killer T cells or cytotoxic T cells kills the antigen, pathogen or cancer cells. Third type is suppressor T cells. You can write it as TS. Suppress करता है. Suppress करने में थामोने. These cells suppress our immune cells. That is B lymphocytes, T lymphocytes, and stop the immune response. Antigen नष्ट झाल्यानंतर स्टिम्युलेट झालेल्या ज्या काही आपल्या इम्युन सेल्स आहेत, बी लिम्फोसाइट्स, टी लिम्फोसाइट्स, विशेष करून किलर टी सेल्स, यांना इनएक्टिव्ह करणं गरजेचं आहे. अदरवाइज ते आपल्याच शरीरामध्ये नुकसान करतील, हार्म करतील, आणि ते अवॉइड करण्यासाठी आपला इम्युन रिस्पॉन्स थांबवला गेला पाहिजे, तो सप्रेस झाला पाहिजे. त्यासाठी त्या सप्रेसर टी सेल्स काय करतात, या बी लिम्फोसाइट्सला किंवा टी लिम्फोसाइटला त्या ठिकाणी सप्रेस करतात, इनएक्टिवेट करतात. त्यामुळे इम्युन रिस्पॉन्स जो आहे तो थांबून जातो. आणि चौथा प्रकार आहे, द फोर्थ टाइप इज मेमरी टी सेल्स. मेमरी टी सेल्स, these are the T lymphocytes which retain memory. Their function is to retain memory of immune response, or you can say antigen encounter. सो ह्या ज्या आहेत मेमरी टी सेल्स, यांचं मेन फंक्शन आहे अँटीजेनचं जेव्हा एनकाउंटर या बी लिम्फोसाइटचे अँटीबॉडीजद्वारे किंवा इतर ज्या काही टी लिम्फोसाइट्स आहेत त्यांच्या साहाय्याने जेव्हा अँटीजेनचं एनकाउंटर होतं त्यावेळेला जो लढा होतो, जी लढाई होते, जी काही इम्युन रिस्पॉन्स आहे किंवा तो जो काही एनकाउंटर आहे, तो एनकाउंटर लक्षात ठेवायचा, रिटेन इन मेमरी. त्याचा फायदा केव्हा होतो? जेव्हा तोच अँटीजेन दुसऱ्यांदा आपल्या शरीरात जाईल त्यावेळेला या मेमरी टी सेल्स सगळ्यात पहिले ऍक्टिव्ह होतील. बिकॉज दे नोज दॅट पॅथोजन, दे नोज दॅट अँटीजेन, त्याचे फीचर्स त्यांना माहिती आहेत, त्यांनी त्यांच्या मेमरीमध्ये ते ठेवलेले आहेत. आणि म्हणून मेमरी टी सेल्स विल गेट सेन्सिटाइज फर्स्ट. दे विल सेन्सिटाइज दिस हेल्पर टी सेल्स. Helper T cells will sensitize the B lymphocytes and killer T cells. And again, second time, immune response will be started. And it will be quick and fast. Now, we have to discuss mechanism of action of B lymphocytes. B lymphocytes are the immune cells which provides us humoral immunity or antibody mediated immunity, and how these cells perform their action, that topic we have to learn now. So B lymphocytes get sensitized or stimulated by invading antigen or helper T cells, and after sensitization, after stimulation, they starts proliferation. Sensitized B lymphocytes divides rapidly, to which we call proliferation, and they produce daughter cells. Those daughter cells are called plasma cells, and these plasma cells produce protective chemical which is called antibodies, and these antibodies gives us protection. These antibodies are, they either remain free in blood, or sometimes they get attached to surface of antigen. So B lymphocytes get sensitized, stimulated, and starts proliferation, produce plasma cells, and plasma cells produce antibodies. Second type of cells of immune system are antigen presenting cells. These cells of immune system, they perform different functions. First, they engulf the pathogen. Along with this, they also carry out processing of antigen. And these processed antigens are carried on their surface, and that's why their name is antigen presenting cells. They also deliver stimulatory signal for helper T cells, or you can say they stimulates helper T cells. So these are the three important functions carried out by these antigen presenting cells. Now we have to discuss the antigen antibody interaction. Antibodies which are formed from the B lymphocytes or their clone cells, that is plasma cells, how they interact with the antigen, how they destroy or kill the antigens, those interactions now we have to learn. The first interaction is neutralization. In this interaction, antibody binds to toxins, viruses or bacteria, means antigens, and block their attachment with host cells. Due to this, infection is prevented. Prevention of infection. It also helps toxic action is also reduced due to this interaction. Second interaction is opsonization. In this interaction, antibody coat the surface of antigens. It forms the coating on the surface of antigens and signals the phagocytic cells like macrophages and neutrophils. Due to this coating of antigens by antibody, these phagocytic cells, they get activated and they start engulfing or phagocytosis of those coated antigens. So we can say opsonization increases phagocytosis. Phagocytosis by macrophages and neutrophils. And the third interaction is called agglutination. Agglutination. In this interaction, antibody binds to multiple separate pathogens or antigens. Pathogens or antigens. And due to that, due to this linking of the antibodies to the multiple antigens, it forms the clumps. And due to the clumping of antigens, they become immobilized. So, immobilization of antigens is caused due to agglutination. So, we can say agglutination causes immobilization of antigens, and those immobilized antigens are then phagocyte or engulfed by the phagocytic cells.